Next speaker, we have Bhavesh Sharma and Nimi Bhattatiry. They will be speaking about DNA methylation of TP53 in cancerous and normal cells.
This summer, my partner and I worked on an analytical research paper that dealt with the DNA methylation of TP53 in cancerous and normal cells. With the help of our mentor, Dr. Ma, and a statistical programming software called R, we were able to analyze the trends and patterns in our data sets and identify the characteristics of the TP53 gene. So what is DNA methylation? DNA methylation is an important biological process, and it's part of normal mammalian development. And after replication, DNA goes through a set of DNA methylation is just one form of post-synthesis modification. It usually occurs on CpG islands or the cytosine and guanine bonds bonded by phosphodiesterase bonds, and it has the potential to suppress the expression of viral genes and specific enzymes such as DNA methyltransferases or DMTs in mammals aids DNA methylation of certain genes. These are just two diagrams that we found, and there are two main components of the epigenetic code, and one of them is DNA methylation, and after that it's histone modification, but we just focused on the DNA methylation of certain tumor suppressor proteins such as TP53. And DNA methylation is the addition of the methyl group to the cytosine, the DNA-based cytosine.
Okay, so where we took this project was we applied it to lung cancer. So it's accepted that variant methylation of CpG islands in the frab region of cancer-related genes is integral to their transcriptional silencing. This provides an alternate mechanism for transcriptional silencing. 5-methylcytosine, which is the 5' region methylated, is mutagenic, which contributes directly to causing transitional mutations, which is MECpG to TCpG. And almost all human genes contain methylated cytosine residue in their coding regions. And methylated cytosine residue has been shown to favor the formation of carcinogenic adducts between DNA and carcinogens.
The main gene that we revolved our research around was the TP53 gene, or tumor protein 53. The TP53 gene encodes a P53 protein, which is a tumor suppressor protein involved in lung cancer. The tumor suppressor is involved in preventing cancer. We found that from reading various papers, and it also has the potential to initiate apoptosis or program cell death if there's DNA damage. And we also found that human tumors contain a mutation deletion of the TP53 gene, so we found that the TP53 gene was an important part of lung cancer. The first paper that we read dealt with the variant methylation of P21, which was another tumor suppressor protein in lung cancer. It dealt with the suppression of the P21 gene, and it also mentioned the activation and inactivation of cyclin-dependent kinases that allowed for the initiation of DNA methylation, but it didn't really go in depth, but it was definitely a big part of this paper. In cancer in general, it's also related to P53-mediated apoptosis and can be used to suppress tumor growth. However, lung cancer specifically, the P21 or P53 protein is silenced by promoter methylation in lung cancers. We analyzed this data, and we found from the experiments with the cultivation of non-cancer cells with P53 using anti-P21 and anti-P53 antibodies. And as shown by the red boxes, there's positives and negatives that showed the expression of the genes, and we found that we had positive results for the methylation of P21 in cancer cells and no methylation in non-cancer cells. And this table is just an interesting table that illustrates the different factors that are involved in the methylation of the gene, and such as age, gender, a smoking history, or the stage of lung cancer. We concluded that the modification and hypermethylation of the P21 protein was important to the inactivation in lung cancers. However, in tumor cells, loss of or the absence of P53 led to a reduced expression of P21.
The second paper we used was P53, a transdominant regulator. The purpose of this paper was to determine whether mutations in the P53 protein would disrupt wild-type P53 transactivational capacity. So the reason behind this was P53 mutations in human tumors are found in the coding regions, resulting in amino acid changes. So how this is applied to lung cancer cell lines were used due to their definitive mutations in P53 proteins. And these cell lines were recipients to wild-type P53. So the data, the table shows a percentage of P53 transactivation in 3 different lung cancer cell lines. You have H23, H1299, and H358. They used the GAL4 P53 constructs, and they were designed with missense amino acid substitutions and deletions found in human lung cancer cells. The entire exon 4, which are amino acids 33 through 125 deleted, and P53 with 8 amino acids deleted, and exon 8 from codons 262 to 269. So analysis of the data shows missense mutants exon 4 deletion and exon 8 deletion of 8 amino acids showed greater than 90% loss of activity. So the thing to take away from all of this is modifications to the P53 protein show significant loss of transactivational activity. So in our data sets used, we gathered data from the IARC, which was the International Agency for Research on Cancer, specifically their database for TP53 mutations. So modification selected was a point mutation, specifically the missense mutation on cancer and non-cancer cells. The missense mutation results in protein becoming non-functional. And what we did was we analyzed the frequency of transversions between different nucleotides. Okay, so green column represents nucleotides before, and blue row represents nucleotides after. So in the first cancer cell line, in red you can see high frequency of G to T transversions. And in the regular cell line, you can see high frequency of G to A transversions. So conclusion, notable changes in chemical structure of thymine, which is the T, is noticed during transversion. And comparison of lung cancer cell lines to regular cell lines shows a high frequency of G to T transversions on lung cancer cell lines, and a decrease of C to A transversions in lung cancer cell lines.
We can apply this research to identify how mutations and different environmental factors such as the smoking history can affect DNA mutations. And linking paper 1 and paper 2 and how the methylation and the transversion of dinucleotides, we can use this to identify the specific methylation of TP53 in regards to lung cancer tumor growth. We'd like to give a special thanks to Dr. Ma because he helped us a lot when we were lost, and the NanoExplorer program because we wouldn't have had this on.